In this video, we will discuss about the H-mRNA based vaccine results against other alternatives. In this H-mRNA, this H represents the hepatitis B surface antigen, and we have seen in the previous video that this hepatitis B surface antigens has the good immunogenicities within the patient, and also this hepatitis B surface antigens can be expressed easily on the cancer cells. So that is the reason they have selected this hepatitis B based surface antigens for their further functional studies. First they check whether they are able to whether there is any change in the RNA scale or not. So in this case what they did is they took the mice model and treat them with this H-mRNA based vaccines, and thereafter they implant a graft of this ovarian cancers, and 30 days after that they use this VVH, or that is virus that contains this hepatitis B antigens that can be expressed on the cancer cells, and thereafter after 30 days they check whether there is a change in the RNA expressions occurs or not between the case where mice is treated initially with H-mRNA and the mice that is not treated with H-mRNA, and they are able to see that most of the genes are upregulated and some of the genes are downregulated. The genes that are upregulated are related to the immune response, dendritic cells activations, T cells activations. These things they have further validated using gene enrichment analysis, where they have seen the pathways that are related to the immune cells activations, such as T cells activations, leukocyte mediated immunity, are upregulated compared to the pathways that are not upregulated. So once validating that at the gene levels, the expressions levels, the change in the expressions are as per the expectations, thereafter they wanted to check the efficacies of their H-mRNA based vaccines. So in this case, what they did is they first treat the mice with this mRNA H vaccines at -29 and -15 days, and at day 0 they inject the mice with this B16 ovarian endograft, and after day 6, day 6, 8 and 10 they induce this VVH, this virus that contains the hepatitis B antigens, and then at day 28 they analyze the tumor microenvironment. So first they check the tumor volume, and they are able to see that tumors are able to suppress significantly in the case where they use H-TVAC or H antigen mRNA compared to the other antigens mRNA. And these things they have further validated using the T cells, activated T cells. They are able to see that maximum activated T cells, that is CD4 plus T cells and CD8 plus T cells, are found in their H-mRNA based vaccines compared to the other alternatives. And once validating that these H-T vaccines are able to function efficiently, they then wanted to find out of the different subset of the immune cells, which immune cells are able to perform these tumor suppressions. So for that they use the different antibodies to suppress the individual immune cells, such as anti-CD4, anti-CD8 and NK, natural killer cells antibodies, and they are able to see that the tumor volumes are able to suppress even when they used anti-CD4 and NK11 antibodies, whereas when they use the anti-CD8 antibodies, the effect of the H-mRNA vaccines diminishes, indicating that it's the anti-CD8 antibodies that plays a role towards these immune suppressions. And thereafter they also validated the endogenous expressions of the T cells, whether the dendritic cells are able to provide the antigens to the T cells and cause the T cells activations or not. So in this case they check the ovarian specific IFN gamma, and they are able to see that these ovarian specific IFN gammas are significantly higher where they use H-mRNA vaccines compared to the individual case where they neutralize the anti-CD8, CD4 and anti-NK11 antibodies. And these things they these things they find out, the similar things they find out in the when they check this hepatitis B antigen specific memory T cells. They are significantly higher in the case where they use H-mRNA based vaccines compared to the other control based. Thereafter they check this T cell suppressions that contains the immune, that also contains the immune checkpoint inhibitor surface receptors such as PD-L1 receptors, and they are able to see that the immune suppressions are minimum in the case where they use H-mRNA vaccines compared to the control, that is saline case. So all these results indicates that this H-mRNA these vaccines functions has a better efficacies and are able to suppress the tumor volume compared to the other antigens that they have tested in their studies. So that's all for this video, and if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.